Welcome to class. Welcome to the cell and beyond. Grab a pen and a book while we dive into the world of biology. In this video today, we'll be defining a microscope. We will mention the types of microscope, okay? Because we have different types of microscope, but I will not exhaust everything in this video. I'll just mention a few, all right? And then we'll state the parts of microscopes and the part of microscope and their functions. Okay, so we'll also state the importance of microscope. So join me and let's journey this lane together. First, what is a microscope? A microscope is a laboratory instrument used to examine objects that are too small to be seen by the naked eye. Yes. Now, there's a branch of biology I called microbiology. Yes, this is a branch of biology that studies microorganisms. Have you ever wondered how microbiologists cope with microorganisms? Okay, the only instrument that makes microbiologists that helps the only instrument that helps microbiologists relates well with the microorganism world is the microscope. So microscope is a laboratory instrument. So we use it in the lab to examine objects. Whatever thing that is too small to be seen with the naked eye, of course, we make use of the microscope. All right, so let's look at types of microscope. We have the compound microscope. Majorly, this is the biological microscope. This is the one we use in the biology lab, okay? And it's a microscope with a high resolution and two sets of lenses providing a two-dimensional image of the sample. Now, have you ever heard of compound microscope? Why is it called compound microscope? The reason is because it has two sets of lenses, okay? And what is the function of this lens is to provide a two-dimensional image. The very first lens will provide a particular image. Then the second lens will produce another image. Okay, so it's like it's producing the image in two ways. Okay, making use of two lenses. All right, so that is the term compound, referring to usage of more than one lenses in the microscope. So that is why we call it a compound microscope. All right, now let's look at the parts of microscope. Okay, now this is an example of a compound microscope. Remember, I told you that there are two major lenses. The very first lens is the eyepiece lens. Can you see the first lens there, where the observer will have to use to view with the eyes? Okay, and the second lens is the objective lens. Okay, so that's the more reason why we call it a compound microscope, because of these two lenses, the eyepiece lenses and the objective lenses. Okay, so that's the more reason why we call it a compound microscope. Now this lenses comes, especially the objective lens, it comes in 4 times 4 magnification, times 10 magnification, times 40 magnification, and finally comes with in times 100 magnification. So you can just switch the objective lenses depending on what you want to view, depending on how big you want the image or whatever you want to view. You want it how big you want it to be. Okay, so that is why it comes in different magnification. That's 4 times, 10 times, 40 times, and 100 times. Okay, that's the magnification. All right, so let's continue. Now essentially a compound microscope is a high magnification microscope that uses two lenses to compound the level of magnification, like I said. Okay, the first lens is referred to as the objective lens and typically has times 4, times 10, times 40, and times 100 magnification ability. So the second lens is known as the eyepiece lens, like I explained earlier on. This is just to expand what I just said. Okay, now let's look at other types of microscope. We have other types of microscope. The very first one, we have the polarized microscope. Polarized microscope. This is how it looks like. All right, polarized microscope. We have other microscope too. We have the stereo microscope. Look how it looks like. This is the stereo microscope, and one is the electron microscope. Electron. This is how it looks like. Literally, you need a room for this. Okay, you need a room for an electron microscope. Okay, so it's a very big one. That is how it looks like. And the next type of microscope is the fluorescence microscope. This is a fluorescence microscope. And another type is the metallurgical microscope. This is how it looks like. You can see all of them are compound microscope. Yes, all of them are, apart from electron microscope. That one is a different one. I'm still going to expatiate on that. Okay, all the ones I've mentioned earlier on are a compound microscope. Okay, because they make use of two lenses. All right, one lens are the eyepiece lens, and the other lens are the objective lens, just to compound the image, just to make the image more pronounced. So that's why we call it a compound microscope, but different types, right? Okay, so if you look at this particular image, now look at this particular image. It is also a microscope. Yes, we also use this, especially in the secondary school laboratory. This is a monocular compound microscope. Now, the more reason why we call it monocular is because the eyepiece lens is just one. Okay, the eyepiece lens is just one, so that's why we call it monocular, but it is a compound microscope. The reason is because there is a lens here at the top, here at the eyepiece, and another lens at the objective lens area. So it's still a compound microscope, but monocular microscope, but only one eyepiece lens. I hope you understand that now. So it has only one eyepiece lens, so hence the name monocular. So take note of that. But the one I showed you earlier on is a binocular. Yes, a binocular microscope, because it has two eyepiece lenses, so hence the name binocular microscope. Now, as a science student, you'll be coming across some prefixes like monocular, like mono, bi, okay, tri. All those things will help you understand what it is. It's all about some words, okay, like bi means two, mono means one, poly means many. Okay, so take note of that. All right, so let's continue. So you can see those two lenses. Let us look at the parts of the microscope. I'll be looking at their um, functions. Okay, help. Let's look at them one after the other. We have looked at the revolving nose piece, the objective lens, the stage, the condenser, the illuminator, the base, the eyepiece lens. I've said that several times, so by now you should be able to remember that. That's the arm. We have the clip. I hope you can see clearly. We have the coarse focus, and we have the fine focus. We can call it coarse adjustment knob, or we can also call it fine adjustment knob. So take a look at this. That's the major part of a microscope. So in the next slide, we'll be looking at their functions one after the other. All right. What is the function of the eyepiece lenses? It magnifies the image produced by the microscope's objective lens so that it can be seen by the human eyes. All right. So remember, I told you that the objective lens will produce an image. And where is the objective lens? This is the objective lens. Okay. Objective lens. All right. So any image produced here, okay, will be magnified here so that the eyes can see it. I hope you understand that now. So that is the major work of the eyepiece lens, to help magnify, sorry about that, to help magnify whatever image that is produced by the objective lens. I hope you understand that. Now let me repeat myself. Whatever image that is produced, whatever image that is produced by the objective lens. And what is the objective lens? This is the objective lens. Alright. So whatever image that is produced by the objective lens will be magnified by the eyepiece so that it will be very easy for the eyes to see it clearly. Alright, so that is the function of the eyepiece lens. Alright, I hope you understand that now. Let's continue. Let's look at the next thing that I want you to understand. This eyepiece lens also comes in different magnification. Okay, it comes in different magnification, so it is removable. You can actually remove this eyepiece lens. You can remove them, of course. You can remove them and change it to whatever yeah, that part can be removed, and you can change it to whatever magnification you want. Yeah, it comes in times 20 or so. It comes in times, I think, in times 10 also, so you can remove them. And it also comes in times 4, like that of the objective lens too, because the objective lens too comes in different magnification, but that of the objective lens is not detachable. Okay, you can't remove them, but the one from of the eyepiece lens, you can actually detach them and place these two images I'm showing you right now. You can easily place them back, and whatever, you can switch it whatever one you want. Okay, so that is that about the eyepiece lens. The next part, the revolving nose piece. Please don't mind this arrow. The revolving nose piece. I hope you can see where the arrow is pointing at. I mean this part. Let me just try to highlight that for you. This is the revolving nose. Okay, this is the revolving nose. Now this part actually is movable. That's why we call it revolving. Okay, it is movable. You can move it whatever direction you want, just to select each of these objective lenses. Okay, is 4 of them, like I said earlier on. You can revolve, you can switch this, you can move it, you can turn it. Okay, you can turn it to select whatever I what to select whatever objective lens you want. All right, this is the part that holds 2 or more objective lenses and can be rotated to easily change power. So whatever power, either the times 4, the times 10, or the times 40, or the times 100, depending on what you want. So it is the revolving nose that helps you to just select it easily. Okay, all right, so that is that about the revolving nose. Okay, so you can see how it looks like here. All right, so you can easily rotate it and select whatever power you want. Is it times 4? Is it times 10? Is it times 40, or times 100? So whatever way you want it, depending on what you want to view. So then let's look at the next part. Of course, the objective lenses. All right, the objective lens is a microscope captures the objective lens in the microscope captures and refracts the light reflected from an object, because I want you to understand something. Every object actually have the kind of light they produce. Okay, because it's the reflection of everything that you have that makes it possible for you to see. That is the light that the eye will be able to, interpret that this is what you're saying. All right, so any object that you want to view, there is a light that comes from it, and the objective lens will refract this light. It will bend the light. Okay, it will bend the light, and that is why it is very easy for you to be able to view it easily at the eyepiece lens. Okay, all right, so even a tiny, even a very tiny image, you'll be able to view it depending on the power you want to use. Okay, remember I told you this power comes in different magnification. We have times 4, we have times 4, we have times 10, we have times 40, and we have times 100 magnification. So they come in different magnifications. So, and you can select any of this magnification using the revolving nose piece. I hope you understand. I hope I'm making sense. Okay, so let's look at the next part. Coarse focus or coarse adjustment knob. Now this one, if you look at it, you can see where it is pointing to. All right, I hope you can see that. I hope you can see that this particular one here, this big one, this one, I mean this particular one. I hope you can see. Okay, let me change the highlighter. Okay, I mean this. I hope you can see the direction of the arrow. Okay, now this particular one, this one here. Okay, now that is the coarse focus or coarse adjustment knob. So you can call it any of that, you're correct. Now it's a rapid controls which allows for quick focusing by moving the objective lens or stage up and down. All right, please, that's a typo error, and down. All right, so now the work of this is just to help move this stage. This is the stage. Okay, this is the stage, to move it up and down, just to make sure that the objective lens have a good focus of the object you want to view. I hope you understand that now. You have a glass slide here, and you want to view something on this glass slide. So it is the work of this coarse adjustment knob which will help you move the stage easily for fast focus. All right, so the next part is the fine adjustment knob. So you can call it fine focus. All right, fine focus knob, whatever one, whichever one is correct. Now look at this small one here. Let me highlight it again, this particular one here. All right, this particular one here, that is the that's the fine adjustment knob. And what is the function of the fine adjustment knob? What is the function of the fine adjustment knob? Now, once the coarse adjustment knob is used to bring the object into a general focus, okay? Remember, I told you the coarse adjustment knob helps to move the stage, all right? It helps to move the stage so that the object can be in a proper focus. This is the stage, like I told you, to help make the object come into proper focus. And what is the function of this? That's when this comes in, the fine adjustment knob. It helps to bring the object into a sharper focus, makes it clearer, all right, by making minor changes, okay, to the stage position. All right, so it gives a minor changes, a minor movement to the stage so that the image can be clear enough to view. All right, so take note, you have to first of all use the coarse adjustment knob before using the fine adjustment knob. The coarse adjustment knob, which is this one, helps to bring the image to proper focus, okay, by moving the stage. All right, then while the fine adjustment knob is to help make it have a sharper view, all right, by just moving the stage slightly or whichever way, just to have a clearer view depending. So that is that about that is fine adjustment knob. Now let us look at the next part. The next part is the stage. Of course, all microscopes are designed to include a stage. Of course, that is where the specimen. Now the specimen is usually mounted on a glass slide. Now if you look at this image very well, you can see like a flat something, a flat glass. Yes, a flat glass. That is where the specimen will be placed on. Now this is a glass slide here. this one here. Okay, there's a glass light. Okay, now that glass light is where the specimens will be mounted on. All right, so in this channel you'll be seeing series of ways that um, specimens can be mounted on a slide. Okay, so that is where the specimens will be mounted on, and it will be placed on that. Okay, so that is the function of the stage. All right, I hope you understand. All right, now let's look at the next part. Now, what part are we talking about? Is the condenser. Now, if you look at it very well, very well, the condenser, they act to gather light from the microscope's light source. Now, where is the light source? Now, look at this image down here. Look at this particular thing here, this one here. That is the light source or the illuminator. Now you can see that it is actually illuminating light. It is bringing out light at this region, and once it directs light directly, it goes to the condenser. So once it gets to the condenser gathers the light from the microscope light source or the illuminator and concentrates it in a cone way, and illuminate it to the specimen. This is the specimen here. All right, so it will gather the light and narrow it down to the specimen so that anyone who is viewing it will be able to see it under a proper illumination. I hope you understand that now. Okay, so let's continue. Then let me show you a clearer view of what the condenser look like. Now this is a clearer view of the condenser. This is the condenser, okay, where this hand is holding, that's the condenser, and this is the knob that the hand is holding. This knob, it helps to gather more light. It's like you may decide to let the lights dim down the light or increase the illumination of the light depending on what you want based on this knob here. We call it knob. Okay, so you find it around the condenser. So this is the condenser. This is the condenser. Okay, all this here is the condenser. All right, and this knob here helps to control the amount of light that enters the specimen. Of course, you know that the specimen is somewhere here, and this is the objective lens. All right, and so whatever light coming from the illuminator here, all right, is directed into the condenser. All right, so the condenser gathers the light and directs it to the specimen here. Okay, so that is that about that. All right, now let's look at the next part. The next part is the illuminator. Where is the illuminator? So I've mentioned it a while ago. That's the illuminator. Is it provides light, transmit light through a translucent object. Okay, some objects, in short, all the objects are, some objects are translucent. It doesn't allow light to pass through it. So but this illuminator helps light to go through any translucent object. All right, you know, when we say translucent object, light passes through it but not completely. Okay, doesn't pass through it completely. Then the next one is the clip. All right, can you see the clip there? Now the function of this clip is to hold the specimen slides in place. I told you that there is a slide that you mount your specimen on, so this will help you hold it in place. Okay, that's the function of the clip. Then the next one is the base. We have the base. The base, what's the function of the base? It serves as a support for microscope. Okay, and again you see the illuminator mounted on the base. Okay, you see the illuminator mounted on the base. This is the illuminator here. You find it on the base. This is the base. So whenever you lift your microscope, this is what gives the microscope support to sit properly on whatever desk you're putting it. Okay, now the next part is the arm. And what's the function of the arm? Is support the tube and connect it to the base. So you can also use this arm to lift it. You can also use the arm to lift the microscope. Now let us look at the importance of microscope. Microscopes allow scientists to investigate cell structure, observe the tiniest details of plants, animals, and fungi, and learn about the presence of microbes. So there are things that cannot be seen with the naked eye. The, the microscope helps to um, elaborate it or escalate it so that we can have a proper view, especially the cell. The microscope helps to view this very well. Okay, so let us recap. We define a microscope. We also state the types of microscope. And we also mention the types of microscope and their functions. So we also stated the importance of microscope. So I have a question for you that you will drop for me in the comment section. What is the difference between a light microscope and an electron microscope? So I'm expecting your answer. What is the difference between a light microscope and an electron microscope? Yeah, there is a difference between the two of them. So drop your answer for me in the comment section. I'm waiting in the comment section. Bye for now.
If you like this video, why not subscribe for more videos? Like and share. Someone needs to see this video. Thank you.